Uh, it's welcome, Anna and Apita. So Austin is not here today. It's okay. So there's PhD students from Anshar's group from Stanford, and they are interested in developing and evaluating robust and biologically motivated DNA sequence models with a particular focus on regulatory DNAs. So today's talk would be about this. I don't know how you guys pronounce your model. Maybe DART-Eval. It's a benchmark framework. So let's welcome them and just please take it away.
Awesome, thank you. Thanks for the introduction. Thanks for having us. My name is Aman, and along with Arpita and Austin, we've developed DART-Eval, which is a evaluation framework for DNA language models on regulatory DNA. Um, and so self-supervised language modeling strategies have proven extremely useful in a wide variety of fields, including text. In proteins, they've had a lot of utility as well. And recently, a lot of works have come out to try to apply them to DNA in addition. And so there have been quite a few of these that have been released recently, including a lot of these works here, plus several more that have come out since this presentation was put together. And these, encompass a wide variety of strategies, a wide variety of architectures, but they all sort of had the same goal of representation learning on DNA sequences in a self-supervised manner. But DNA poses some unique difficulties that we need to navigate for producing effective regulatory DNA models. The first, that DNA is extremely heterogeneous. So there are many different sort of types of DNA which can be considered unique languages. And an effective DNA language model would need to account for all of these, with the problem being that the actual functional DNA is so sparse with respect to what we call neutral DNA, or DNA that lacks any kind of obvious known function. So these are interspersed with each other, and there's no clear pattern to when one starts and the other ends without external annotations. And so this sparsity and the heterogeneity of DNA makes, in theory, DNA language models very difficult propositions to get correct. And also, so regulatory DNA especially is something that is an extremely important type of DNA to get right. And the syntax of regulatory DNA here is it's cell state specific, where the same stretch of DNA can be read in very different ways depending on cell type, due to the presence of different transcription factors, cofactors, etc. And this context specificity makes regulatory DNA extremely expressive, but it also poses some really unique challenges to any attempts to model it. But unfortunately, the rate of model development that we have has greatly outpaced the ability of robust evaluations, especially for regulatory DNA, where it's essential to evaluate the full expressivity of the models on this very complex data type. But what we argue is, unfortunately, the evaluations just currently are not there. So first of all, the benchmarks that exist in literature often involve data sets or models that are well behind the current state of the art. So the field has progressed in general massively in the past few years outside of the self-supervised paradigm, and a lot of language model evaluations haven't really caught up with that yet. So when you're in terms of the models and the data sets they're comparing to, they're not really getting a good gauge on whether the DNA language models are actually competitive with the current state of the art in the field. So that needs to change. And also, there are some serious data set design issues with these benchmarks. For example, a lot of benchmarking tasks involve the comparison of positives to negatives, and the negatives have to be chosen robustly. Otherwise, you get caught up with confounders, and it's not really a very useful task. And I think that a lot of the negatives that are chosen in benchmarking tasks in the literature have not really been given this kind of thought. So the task that you have is not really one that's too biologically useful. There are a lot of other problems, like, for example, when you look at variant effect prediction, which is a very important task where you look at what are the effect of genetic mutations. You have to be really careful in your ground truth variant set to account for things like linkage disequilibrium, or spurious correlations and things like that. And a lot of tasks do not do that. So it's not really hard. It's not really easy to kind of figure out how well the model is actually gauging variant effects. And arguably the biggest problem is that a lot of these benchmarks only involve surrogate predictive tasks that don't really address key downstream applications. Because if you think about what you might actually want to use these models for, it's things like maybe revealing sequence motifs or finding variant effects or designing synthetic sequences or something along those lines. And whereas a lot of these benchmarks are only evaluating on the actual classification tasks that the models have been fine-tuned on, which doesn't directly tell you how well it's going to do with these actual downstream tasks that biologists care about. So in conclusion, there are a lot of issues with the existing DNA language model benchmarks out there. And what we hope to do is release a suite of tasks that helps you make more informed evaluations of these DNA language models. And so to fill this gap, we introduced DART-Eval. So this is an adaptable benchmarking framework, which we'll go through each task in more detail. And it consists of a series of tasks in increasing difficulty that assess a wide range of regulatory DNA use cases for DNA language models. So let's now go into detail about the approach we're taking here. So we're applying our evaluations on 6 prominent and recently published DNA language models, which are listed here. These encompass a wide variety of architectures, a wide variety of context sizes, some different training strategies in there. Some are only trained on the human genome. The others are trained on human and mammalian DNA. So there's a lot that's out there, but the common denominator here is that they train self-supervised models on a genomic level. And then for each task, we have some combination of zero-shot and supervised evaluations. And in the zero-shot space, we could look at things like embedding distances or the likelihood that's calculated for a particular sequence. And in the supervised setting, we do either final layer probing or fine-tuning using LoRa. And these are all meant to mimic ways in which people might use the models in their own settings for adapting to specific tasks. And for each task, we have pretty rigorous and high-performing baseline models that are basically ab initio or trained from scratch. And these are typically dilated convolutional architectures, which have been shown to do really well for a lot of supervised genomic tasks in the past. Okay, so let's now go into the tasks that we're working with here and the results that we found. So starting with our very first task, which again is the easiest, and you can see that by the number of chili peppers up here in the top right of the screen. So this task is the easiest and involves testing whether the models can basically detect any kind of regulatory DNA. So this is like the sort of very lowest level of difficulty that we can potentially expect. And on a high level, what it involves is comparing regulatory sequences to controls and determining whether the models can tell those apart in zero-shot and supervised settings. So using likelihoods and then using classification probabilities. And to go into this more detail, the positive examples in our data set consist of 2.5 million candidate cis regulatory regions as defined by the ENCODE consortium. And you could think of these as basically high confidence regulatory regions. And our negative set consists of these sequences, but shuffled in a way that some important sequence properties are preserved. So these include things like the GC content and also the frequency of dinucleotides, which are 2 nucleotides in a row, basically. So this kind of shuffling means that these sequences are basically compositionally equivalent when it comes to those confounders. And any classifier that you train on them wouldn't just be able to pick up on those confounders and produce good results accordingly, which has been a big problem with the previous benchmarks. So the only difference between the two categories is the presence of, real functional regulatory motifs in the positive set, which is what we expect the models to detect. So we test this in 2 ways. In the zero-shot setting, we look at the model likelihoods, and we expect the basically according to the reconstruction probabilities. And we expect the models to assign a higher likelihood to the regulatory sequences than to their negatives. And in the supervised setting, we can pretty simply train a classifier to differentiate the two classes. So first of all, our supervised CNN baseline proves pretty effective at discriminating the two categories, getting over 90% accuracy. And if we look at a zero-shot setting first, we see that our Caduceus model, for example, is pretty competitive with the baseline, as are the other models, although slightly less so. And then when we get into the supervised settings, the DNA language models start to outperform the ab initio models pretty regularly. So overall, we're seeing strong accuracies across the board for this task. And I would argue that these results generally reflect the lack of difficulty that this task has. And we see this as kind of the bare minimum or the preliminary bar we have to clear before getting to the more complex evaluations. So speaking of which, the next task that we have is intended to see if the models can graduate from recognizing any type of regulatory DNA to specific regulatory motifs, right? Because the more of those that they're able to recognize, the more effective they are in practical settings. So we restricted the zero-shot setting in this case, and the way we do this is we use a list of 1,443 known transcription factor binding motifs from the Hokomoko database, which is one of the most commonly accepted ones. And for our neutral background sequences, we have 100 of the dinucleotide shuffled negative set from the previous task. And basically for our positive set, we take these motif sequences and we individually insert them into the center of each background sequence. And then for a negative set, we do the same thing, except with versions of these motifs that are shuffled and therefore likely inactive. So once again, the idea is to see if the model can differentiate active motif regions from inactive regions, but this time at the specific motif level. And we can see how many of those it gets right. Okay, so let's take a look at Caduceus again, and specifically in the likelihood setting here. So here we define an accurate prediction in which the model assigns a higher likelihood to insertion of the true motif than to the shuffled one. And then we see here that in this plot of per motif accuracies, we see the model performance is generally effective, but not perfect. So the plot here is showing the CDF basically for the accuracy for each motif. And the line here is just for visualization purposes, is that 70% accuracy. So we can see that for around 20% of the 1,000 or so motifs that we're working with, they exceed the 70% threshold. So the models do identify a good number of motifs pretty well, but not all of them for sure. And there is a large number for which the accuracy is very poor. But still, this is a slightly promising result. And these patterns are generally carrying over to the other models as well, where some are better than others, but all of them show reasonable performance over a certain degree, with poor performance in some motifs. However, the last layer embeddings of these models, and this is going to be a theme that we sort of carry through the rest of the presentation, they seem to show very little information, if at all. So here we're defining an accurate prediction in which we take the neutral background without any insertion in it. We calculate the embedding, and then we calculate the embeddings of the insertion with the true motif and with the shuffled motif, and we see if the distance from the neutral to the positive sequence is greater than the distance from the neutral to the negative sequence, because they should be more functionally different. But as you can see, all the accuracies seem to hover around 50%, which is not informative at all, and it indicates that the embeddings are not really capturing any kind of functional information about the motifs, the regulatory motifs that it should be learning. So overall, very mixed results on this task, where we see some promise with the likelihoods, but almost nothing really with the last layer embeddings. And then so now Arpita can take over. Probably best if she shares her screen. So I'll stop sharing.
Is the next speaker there?
I think she She just left the meeting. I'm not sure what happened.
Some technical difficulties, maybe. No, like what happened last time was the speaker had to enable screen sharing permissions from their end. So maybe something similar happened.
Okay. I guess while she figures that out, you guys have any questions on the first part of the talk?
It's a lot to take in. I'm still processing it, but by the end of the call, I'll surely have one.
Okay.
Well, I had one off the bat actually, but the choice that you had like 6 models and they're mainly like sequence based, right?
Yeah.
There are other like genome language models that have more like supervision tasks, like Informer or Borzoi. Have you thought about maybe, or is that just not a fair comparison? But if you were to incorporate those kind of models, you might see differences, right? Or would you? I guess that's my question.
Yeah. No, I mean, I think definitely. I mean, Informer and Borzoi are, specifically geared towards epigenetic and gene expression predictions, and so I mean that's definitely a very relevant class of models. But I think for this this purpose and these comparisons, we were specifically gearing it towards self-supervised models and all the things that I guess it's claimed that can be done with them.
Right. Okay. No, I'll have another question, but I could save it for
okay.
Later. But then our Pita just left again. So
okay, go ahead. We can.
Sorry, I was just having some technical difficulties with sharing. Is it working or? Okay.
We hear you, yeah. And yeah,
okay.
we see your slide.
Are you guys able to see the screen?
Yes.
Okay, perfect. Sorry. Okay. And it hopefully is just in slideshow mode.
Yeah, it is.
Okay, great. Okay, so yeah, okay. So just moving on from the previous task that Aman presented, now we're going to be focusing on identifying specific transcription factor motifs. And in order to identify those, it's also important to understand cell type specificity. And so, as we said earlier, a hallmark of non-coding regulatory DNA is its cell state specificity. So it's important to evaluate whether models can learn these like cell type specific regulatory vocabulary. And to achieve this task, we use chromatin accessibility peaks measured by a TAC-seq for 5 cell types that come from very different regions of the body. So we have these lymphoblastoid, liver cells, stem cells, lung cells, and leukemia cells. And each of these different cell types also exhibit very different accessibility patterns, as we can see here in the pink. These are the highlighted differentially accessible peaks per cell type. And moreover, each of these sets of differentially accessible peaks contain distinct regulatory DNA, including specific motifs that characterize them. So using tools like Homer within these accessible peaks, we identify these specific motifs where we see high expression of just these motifs in these cell lines and not in the other cell lines. So you would think it's relatively easy for the models to identify these cell type specific motifs or cell type specific peaks when given all of them. And so, we asked the question, can models learn cell type specific regulatory DNA using zero-shot embeddings, where we first evaluate models using model embeddings that are produced on differentially accessible peaks in each cell type. And baseline embeddings are produced by featurizing each peak as a vector of motif counts, and then we cluster all of these different embeddings using k-means. And we evaluate the embeddings both visually and quantitatively. So first, visually, we look at the UMAP of the embeddings of these sequences. So here we can see the bag of motifs baseline approach visualization. And quantitatively, we perform a k-means clustering and use mutual information to evaluate the clustering quality with respect to the true labels. So here we can see that the bag of motifs baseline approach is able to identify modules that can separate by cell type. And now, again, as an example, let's take a look at caduceus. So here, the model embedding clusters are not separable at all by cell type compared to the baseline embeddings that we see. And furthermore, Furthermore, the mutual information scores support what we see, with the caduceus embeddings having a much lower score compared to the baseline embeddings. And this trend again continues for the other DNA language models where they're unable to classify cell types similarly to caduceus, and they perform much worse than the baseline, both visually and again, quantitatively. And okay, so we see that the models don't perform on par with the baseline in the zero-shot space. But what if we help the models using supervision? So this time, we will train a 5-way classifier on chromatin accessibility peak sequences coming from each of the 5 cell types. And we measure the performance of the classifier using AUROC. And as we can see, the ab initio baseline is able to classify cell types well, with an AUROC of little more than 0.9. So what about the DNA language models? We see, again, caduceus in the supervised probed model underperforms the baseline at a much lower AUROC than the ab initio. And so do the other supervised DNA language models, whether they're in probed settings or fine-tuned settings. So overall, we can see not great performance, at least for this cell type, for GM12878. And moving on, we also see that they perform similarly for other cell types. So generally, the models perform barely on par with the supervised baseline, even after these much larger models are fine-tuned. So next, we want to evaluate whether models can quantitatively predict regulatory activity, which is an even more difficult task. So chromatin accessibility is a strong quantitative indicator of a sequence element's regulatory activity. And if a model can successfully provide a quantitative measure of accessibility from sequence, the model can be used to provide a more nuanced understanding of non-coding cell type specific regulatory syntax. And so this is a very common classical task for which many supervised models already exist. But now the question for us is, can we do better with DNA language models? And we evaluate for this using chromatin accessibility peaks from the same 5 cell lines that we used in the previous task, along with their GC-matched inactive sequences. And so we benchmark the models using supervision, where we train models on the peak sets and corresponding negatives to predict the log DNase read counts over the input regions. And we evaluate model performance using Spearman correlation between the predicted and true peak counts. And here we see that the supervised sequence to activity ab initio, or trained from scratch model, is represented by the dashed line, performing at a Spearman correlation of about 0.55, as you can see here. And the random Spearman correlation is at 0. And then the sequence to activity caduceus underperforms the ab initio model, again, in both in the probe setting, as we see here, at quantitative regulatory activity prediction. And we see that, again, it also underperforms in the fine-tune setting. And we see this again for other sequence to activity DNA language model based models, with both the probed and fine-tuned performance well below the ab initio performance. And so this is not much of a surprise, since task 4 is definitely harder than task 3. And we saw that they didn't perform as well as a baseline in task 3, but this is still an important task to evaluate. And we see that this occurs across, again, all cell types, where this supervised probed and fine-tuned DNA language models perform barely on par with the ab initio baselines. So what we see typically in previous evaluations and benchmarks, they end at this surrogate task. But we cannot just rely on observational performance when evaluating models. It is necessary to evaluate models on true counterfactual tasks, such as our next one. So in our next task, in task 5, we benchmark models on this counterfactual task where models are evaluated on sequence they have never seen before in their training set. This task tests whether sequence to activity, or S2A models, can predict effects of genetic variants on regulatory activity. This is the most difficult and biologically meaningful task in our benchmark. If models can accurately predict variant effects, we can determine how a single allele is influencing the regulatory readout. And to test these models, we use a dataset of chromatin QTL variants present in African and Yoruban LCLs, and utilize our sequence to activity models from the previous task, which were also trained on GM12878 LCL regulatory activity. And so to benchmark our models, we use these rigorously curated variant sets, the African LCL CAQTLs and the Yoruban LCL DSQTLs. And so here the causal QTLs are defined as those that are statistically significant and located within peak regions, thus drastically reducing any confounding effects of linkage disequilibrium. And the control set consists of statistically insignificant QTLs that also fall within peaks. So in order to evaluate the models, we first evaluate them in a zero-shot setting. So for the zero-shot setting, we measure the cosine distance between the embeddings of the reference and alternate alleles, with causal variants expected to produce higher embedding distances. And now to take a look at the performance, we see here our ab initio model is a sequence to activity baseline model from the previous quantitative prediction task. And it's very important to note that these models, including the ab initio baseline, were not trained on any variants directly. They were only trained on predicting the regulatory activity readout. And in order to measure performance, we use AUROC. And as we can see here, the baseline performs quite strongly on both data sets, with almost a 0.8 AUROC for the African LCL QTLs and almost a 0.9 AUROC for the Yoruban LCL QTLs. And to compare with the other models now, so first, again, to take a look at caduceus, we see that caduceus zero-shot accuracy is well below the ab initio at about 0.5 AUROC. And again, we see that other models follow suit. They perform similarly across both data sets in the zero-shot setting with an AUROC well below the baseline. But again, this is a zero-shot setting, right? So why don't we try to help the DNA language models out again using supervision? And what we do here is we use a sequence to activity DNA language models that were trained on the GM12878 LCL chromatin activity from the previous task to predict variant effects. And to reiterate, these are not trained directly on any variant effect prediction. And here we compute the absolute predicted accessibility difference between the two variant alleles and use that as our metric. And so, unfortunately, supervision on chromatin accessibility does not help DNA language model performance either. The DNA language models display significantly inferior performance to the ab initio models across all settings. And so this demonstrates the necessity of real counterfactual tasks to determine true performance of the models. And so that ends all the tasks, our evaluation framework. And so I'll pass it on to Austin, who's here for the discussion and conclusion parts. And we can maybe answer any questions at the end.
Okay, yeah, so hi everyone, I'm Austin. Sorry I wasn't able to join earlier, but I'll wrap up the presentation and I'll help answer any questions. So let me just share my screen. You guys can see everything, right? For me? Okay, sounds good. So yeah, so just to recap, we evaluate state-of-the-art DNA language models in zero-shot, probed, and fine-tuned settings across increasingly difficult regulatory DNA evaluations. And we find that although DNA language models excel at the simpler tasks, their performance degrades with increasing complexity relative to more resource-efficient ab initio baseline models of regulatory DNA that are trained from scratch. So just to summarize, let's recap our key contributions. So we package all of these analyses into our adaptable DNA language model benchmarking framework, DART-Eval, which provides 5 families of biologically motivated tasks and data sets that evaluate diverse aspects of regulatory DNA. And that being said, there is a whole world of other aspects of regulatory activity that we have not touched. And these include long context effects, which we can infer from histone ChIP-seq and Hi-C, and also comparisons of regulation across species going beyond humans. And lastly, other types of functional elements, such as UTRs and splice sites. And finally, it's clear from our experiments that these DNA language models have to get better before being practically useful. And we argue that the unreliable performance, it's really a data quality and an alignment issue in that the noisy and imbalanced whole genome pre-training data, it's noisy and imbalanced. So the lowest hanging fruit, even before really optimizing the architectures of these models, is to improve data curation and training strategies. And some of these strategies that we propose include training local short context models on specific classes of functional elements, such as promoters, 3' UTR encoding sequences, and also possibly adaptive sampling of elements to account for the severe class imbalance that is naturally present in our DNA. And lastly, explicitly conditioning on some sort of cell state context information, because these regulatory elements tend to be cell state specific. For example, a liver cell and a brain cell will have very different sets of active transcription factors and therefore active sets of binding sites. But note that all of these strategies depend on some sort of annotation beyond just the whole genome sequence. So in order to really execute on these sort of strategies, you need some sort of functional annotation that comes ultimately from experimental data. So in a sense, you can argue that such a model is multimodal to some degree. And lastly, it is an open question whether universal DNA language models can be built without the help of these annotations from just a pure sequence standpoint. So yes, so thank you all, and we are thankful to our group, the rest of the Condagia lab, and our funding sources. And you can find a link to our paper here in this QR code. So yeah, thank you everyone. So happy to answer any questions.
Thank you, Arpita, Aman, and Austin for the wonderful presentation. And do we have any questions in the room, Dylan?
Well, I'm alone in the room and I have questions.
Okay.
I do, but I also saw there were questions in the
in the chat. We have
so,
questions in the chat.
And I've had the chance of asking my own question before. So so this is the first one from the Sinu. Maybe Sinu, you want to ask it yourself?
Yeah, thanks. So I'm just wondering, like for across all the tasks, like the ab initio model, they cannot do the zero-shot setting, right? So for example, if the foundation model have some non-trivial performance in the zero-shot setting, even they cannot outperform the ab initio model, they still mean something. Like they can do it zero-shotly.
Yeah, so for the zero-shot setting, it's basically for the ab initio, it's supervised, but it's only all these models are only supervised on regulatory activity readout, right? They're not, even the ab initio model is not trained on any sort of like variant effect prediction.
Oh, okay. Thanks.
Yeah, so yes, and just reiterating that I think our zero-shot evaluations depend on the model being self-supervised. And because our ab initio models are only supervised, they're not arguably as applicable to these zero-shot tests. Yeah.
Excellent. Then there's also a few questions from Professor Yue Li. I don't know if Professor Lee wants to ask them himself, or we could have the presenters answer them directly. It's about tasks 2 and 3 and about evaluating on generative tasks. So Any one of you three, I guess, could pick that up.
Yeah, sure. So the first one says for task 2, were the embeddings averaged over the nucleotides to represent each sequence? If so, the long sequences may dilute the motif signals. The answer is yes, they were averaged. But I would say to that, the analog for this task in in a practitioner setting would be to take a sequence and see if it has regulatory activity or not, I guess, based on the embeddings, right? And in that case, you wouldn't know where the motifs were or how many there were, so it would be difficult in practice to target the embeddings to a particular locations. So ideally, you would still want the representations of the sequence, and I know averaging embeddings is a common way to do it. You would still want the sequence representations to be able to capture the fact that one sequence has functional DNA, the other one doesn't. And for task 3, sorry, I missed it. What is the ab initio baseline model again? For each task, the ab initio baseline is a dilated convolutional model, which is supervised according to whatever task is present. So in this case, it's a model that would classify sequences by what cell type they're active in.
Yeah,
And
so it
third
would be like the 5-way classifier
Yeah.
for task 3 specifically.
Yeah. And then the third one, I can open it up to you guys as well. Any thoughts how to evaluate generative tasks?
Yeah, so I think that's actually pretty hard because ultimately for an evaluation, we want. So for generative tasks, I assume that you guys mean like sequence design, right? And we need some way to essentially validate that this design sequence is real or not. And ultimately, that can really only come from experimental data. I think possibly the closest thing we have to evaluating these generative tasks is to essentially do what we're currently doing, is given a sequence, what is the likelihood that the model evaluates the sequence as, and that serves as a proxy of like the probability that it comes up if you were to run this, if you were to sample from the model, if that makes sense.
Yeah, I would say it's like similar to perhaps like what we do in like task 1 or task 2, where we're just like trying to identify whether the models can see whether it picks up what a regulatory element is, against what's not. So that could be like a basic evaluation of these generative tasks. And then probably for more functional evaluations, you would need some experimental setup, as Austin said. Yeah.
So
Very great.
that finalizes that. I'd like to share a few comments that may or may not finish off in a question, and maybe a brief discussion, right? But to me, it's always surprising to see how when you move beyond the protein function proxy, the structural proxy that has worked perfectly for LLMs and that autoregressive objective, and given like the breakthroughs that we've seen, to me, it's always astounding to see how at the transcriptome level or genomic level, we don't really have that sort of like strong predictive power from LLMs, right? So I guess that's the state of affairs right now in the field. And if you guys were to take the ab initio models with all the information that these LLMs are somehow implicitly encoding, or at least that's the promise, and you had to merge the strengths of both to have a model that's actually better than the sum of their parts, what would you take from the impressive results of the ab initio models, and what would you try and do to, quote unquote, maybe this is unfair, but salvage all the training that went into the LLMs, irrespective of their training objective, even if they do some sort of self-supervision or anything broader, right?
I would say one thing that you could potentially do is to see what happens when you initialize the ab initio models with the LLM embeddings instead of the raw sequence representation. So what we typically do in the ab initio models is just do a one-hot representation of the sequence that you're training on or you're predicting on, and you put that in. But it would be interesting to see that, what happens if you replace the one-hot with the embedding produced by the language model instead. Because as you said, there is some level of signal that's being learned there. It's unclear how ultimately useful that is, and I would argue it's definitely not worth the massive cost that's been put into training. But yeah, I think that's something that could be potentially useful for that. So that's one way to go about it. But I think I just want to reiterate the point that regardless of how, like, what you try to salvage from these models, I think the key right now is to focus on shorter context, and if you're going to do longer context, include as many annotations as possible from epigenetic data or like whatever else you might have, right? Because that's what's really going to help the model target itself towards the regions that are actually functionally important. If you do that, right, then that could produce, I would argue that could produce an even better self-supervised genome-wide language model. Again, not sure if it would be the state of the art or anything, but then you could take those embeddings and you could potentially use them for something even more useful.
I like that answer. Thank you very much.
Hi. So, what do you think about these foundation models for DNAs, or more specific for regulatory DNAs? So, do you think it's still promising for researchers to do foundation model right now, or you expect some other stuffs should be going on like in the moment?
Yeah. So I think I could answer this one. I think having a good representation, like a universal representation of DNA, which is what the foundation models seek to do, I think that is definitely a very useful objective. As in, like, if a model is able to do that, that would definitely be highly useful for researchers. I think it's more so. It's not really the concept of a DNA foundation model that we find flawed. It's more the specific execution so far, like the specific models that have been released so far. And ultimately, as we said, we think that it's just due to the nature of how regulatory DNA is structured in a genome. It's very sparse, very imbalanced. And we do think that if you are able to sample the data more smartly, and or you incorporate other kinds of data, I think these models could potentially be much more useful. I think we do think that there is definitely a lot of room for improvement, and that getting a useful model is definitely feasible.
Yeah, and just to add
Okay.
on very quickly, I think it's just important to make sure that the actual functionality, the benefits of these models are like commensurate to the size, because right now we kind of see that these much smaller ab initio models are able to give us much better performance. Obviously, you need a specific supervised model for each context, whereas these bigger foundation models are supposed to be applicable across all contexts. But while creating these models in the future, I think it's important to make sure that we're not just focused on long contexts or like making really large foundational models. It's important to make sure that these models actually, it's important for them to be even that big in order to kind of be that functional, basically.
Great. I just wanted to piggyback off of Austin's closing comment a little bit, and maybe we can discuss for a minute or two. But that's a great sort of perspective opening question. Sampling versus better data sources. If you had to put your money on one for the short term, where would that be? Better sampling regimes, or just get everybody to rethink biology a bit and work on that data curation?
Well, sorry, you can go ahead.
Yeah, sorry. I was just going to say, if you're, talking about a self-supervised genome language model, I don't know how much better you can get than the human genome, or like some of these models have already incorporated, like, lots of other mammalian genomes as well. So in terms of DNA, in terms of the quality of the data, like, I don't think there's, too much improvement on that side. I would put my money on sampling for sure. Either, just like completely restricted to certain types of regions, or at least heavily upweight them or annotate them in some way. I think that could get you the best gains.
I would agree.
And yeah, and just to add on that, if you include if you loosen your definition of data to also include non-DNA data, for example, like experimental data, chromatin accessibility, ChIP-seq, then sampling and data curation are kind of related in a sense, because they're both dependent on other data sources. And I would so as like a first order, like the lowest hanging fruit is better sampling of DNA, but it is possible that like a multimodal model might work better than just a DNA-based model. Just speculation, like a semi-supervised model or some other kind of self-supervised model that incorporates not just DNA data. Because these experimental data, they do hold a lot of functional information. And it would be a little sad not to use them.
Yeah, we already like have these huge repositories of annotated like data sets, so it's just a matter of using them while training these models.
Yeah, no, that's a very exciting answer, and one I would most definitely agree with. It really sets the stage for what's to come, or the next frontier of foundation models, right? Very, very exciting stuff. Any other questions from the audience? Closing comments, suggestions, sponsorships?
Lottery tickets.
Yeah.
I definitely have a few other questions. I'll ask one. It's quite of a broad question, so let's see if the question makes any sense. So clearly in tasks, I would say, I think like maybe 2, 3, or 3 and 4, you try to tackle like the cell type specificity of the embeddings, and basically demonstrated that naively using the language, the genomic language models don't really work. They're sequence-based, so maybe they're not really catered towards cell type specificity, right? But and then you have the results to back that kind of claim. But more qualitatively, or just like if you had to redesign these models, what accounts for the lack of cell type specificity specifically? So you've shown that it's also not great for the functional readout, so like the counts prediction from ATAG data. So that's one challenge. And perhaps another challenge is the cell type specificity, which kind of are intertwined, but specifically for cell type specificity, what do you think is a challenge there? Would a minimal amount of additional annotation be helpful, which you've done implicitly, but I'm trying to figure out, or trying to, I want to get your opinion on that, basically.
Yeah. So the biological basis behind the cell type specificity is that different regulatory motifs are active in different cell types, and so the sequences that we were specifically using for task 3 are DNA sequences that are regulatory in nature and differentially active in one cell type versus the others. And so if a model can't make that classification correctly, all that means is that it's not learning the relevant motifs correctly. And so it should be able to produce representations that basically show you that, all right, well, this sequence contains this, and this motif, and then this other sequence contains motif X, Y, and Z, right? And then given that, it should be able to sort of cluster those sequences separately, which it's not doing. So that's the biological basis behind why it's not learning that. And I mean so I think just the way to improve that is just to make sure that the models are more effective at learning motifs, especially in the embedding case. And as you saw in task 2, that was not the case at all. They were extremely ineffective in the embedding setting at learning pretty much any kinds of motifs. So that's just the same classic answer that we've given in other questions, which is just include annotations, restrict to regulatory regions, all of that stuff, which will help you target it more towards the regions that are actually functional.
So would you think, for instance, like if I think about the nucleotide transformer paper, they use sexamers, right? To then tokenize the representations. But motifs might be a bit longer. I don't know, 11 base pair nucleotides, or 7, 9, whatever. So my question was also about, like, do you think we just have like a different engineering tricks for this to work better?
I highly doubt it. I don't know if there'll be too much of a difference between using K-mer representations and single nucleotide for something like this. I think the biggest issue is just that regulatory DNA is so sparse in the genome compared to the other non-functional parts of it, and it's it's not only very infrequent, but it's also a self-supervised model without any annotations would be actively confounded by the rest of the genome. That's in a completely different syntax basically, and so it's just kind of washing out the relevant parts. So I think if you just target it to the relevant regulatory regions that matter, I think that makes a world of difference automatically. So I don't think the tokenization or the architecture or anything like that are actually the problems here. And I think if you look in that direction first, I think you're sort of misprioritizing what the actual issue is.
Right. Okay. Great.
Wonderfully put. Well, here we have no more questions in the chat. No more questions from the audience, it seems. So yeah, I would like to thank our speakers one more time. Thank you, Aman, Austin, or Peter, for this. And this finalizes our session, or 8th session for the multi-omics reading group. Thank you very much. That stops the recording.
Thank you.
Thank you.